Assalamu alaikum everyone. Today we are going to present a comprehensive overview of mRNA-based COVID-19 vaccine. Vaccine is a biological substance that protects humans from infectious disease by utilizing body's own immune system. There are several types of vaccines: live attenuated vaccines, inactive vaccines, toxoid vaccines, nuclear acid vaccines like RNA and DNA. What is mRNA vaccine? It's a new type of most promising vaccine technology that is proposed at 1990, and the first dose was given to human at 2009. Its mechanism is quite different from the conventional one. In the conventional type, a weakened germ was put inside the body to trigger the immune response, while in the mRNA vaccine, genetic material in the vaccine instruct our body to populate harmless viral protein. mRNA-based cancer vaccine is one of the examples. Now, what is mRNA? mRNA is a single stranded genetic material that is translated into protein at ribosome by the. There are two forms of RNA vaccine: conventional mRNA that encode antigen of interest. On the other hand, self-amplifying mRNA-based vaccine. It differs from the conventional one as it includes with the genetic replication machinery. It is engineered to make more copies of itself once delivered into cell. This means only a small initial dose is needed. For delivery vehicle, sometimes naked mRNA can be taken up by the cell, but generally lipid nanoparticle or other type of particles used to deliver as a delivery vehicle. As we know, RNA vaccine use mRNA inside a lipid membrane. Looking into step 1-2, we can see that this fatty cover both protects the mRNA when it enters the body and also helps it to get inside cell by fusing with the cell membrane. At step 4, NRM is translated by the ribosome, wherein SAM immediately translated by the ribosome to produce the replicase machinery necessary for self-amplification of the mRNA. We can see at step 6, self-amplified mRNA are then translated by the ribosome to produce protein of interest, which is undergoes subsequent transitional modification. At step 7, the expressed protein of interest are generated and secreted. This mRNA typically lasts for few days, but in the time, if sufficient antigen is made to stimulate an immune response, meaning immune memory in the form of B and T cell is achieved, then vaccine materials are broken down. Now I am giving the floor to Nafisa.
Hello everyone, I am Shams Nafisa Ali. Continuing from Shubho, now I would like to focus on the COVID vaccines that have been composed of nucleoside modified mRNA. As of February 2021, there are only two mRNA vaccines that are deployed against COVID 19. The first one is the Pfizer BioNTech COVID 19 vaccine. It is also known as COVID 19 mRNA vaccine BNT162b2 or BNT162a. The second one is Moderna, codenamed as mRNA 1273. Very recently, a third mRNA based COVID 19 vaccine, CVN CoV, is in the news for being in the late phase of testing. In collaboration with Pfizer, BioNTech Corporation has developed the Pfizer BioNTech COVID-19 vaccine. It is the first COVID-19 vaccine to be authorized by the regulatory boards for using in emergency basis or using as a regular one. This vaccination procedure is pretty simple with two doses given 3 weeks apart via intramuscular injection. In clinical trials, it has demonstrated an excellent efficacy over 95% within 7 days of a second dose. Now, let us introspect on the brief timeline of the development of Pfizer BioNTech COVID 19 vaccine. The work began by BioNTech as early as January 2020. After collaborating with Pfizer in March, they conducted a three-phase clinical trial in USA with 44,000 participants. Then, in November 2020, FDA gave approval to the use of vaccine in emergency cases, following which many countries, including United States, Canada, and Mexico, have authorized its use for regular purpose. In 2021, it is expected to go on mass production with an aim of producing 1.3 million doses and participate in large-scale distribution. The vaccine technology has been developed on the basis of nucleoside modified mRNA or modRNA, which encodes part of the spike protein found on the surface of the SARS-CoV-2, triggering immune response against infection by the virus protein. In addition to the mRNA molecule, the vaccine contains some inactive ingredients or excipients. They include lipids, cholesterol, inorganic salts, sucrose, water, etc. But how does the modRNA differ from a standard mRNA? The modRNA sequence is different from mRNA as it does not contain any uridine residues. The residues are replaced by 1-methyl 3' pseudouridylyl. There are also two proline substitutions that cause the spike to added a perfusion stabilized conformation, reducing the membrane fusion ability, increasing expression, and stimulating neutralizing antibodies.
Thank you for the stage, Nafisa. I am Ragib Abid. Along with Pfizer, another American company named Moderna developed an mRNA-based vaccine with the collaboration of Biomedical Research and Development Authority (BARDA) and National Institute of Allergy and Infectious Disease (NIAID). The mRNA present in the vaccine is nucleoside-modified mRNA called mRNA 1273. It encodes the S2P antigen protein. It is simply a modified spike protein containing two proline substitutions in the 986th and 987th amino acid position. This causes the protein to stabilize in its perfusion conformation and adopt a shape that stimulates the neutralizing antibodies. Let us go over a brief timeline of the development of the vaccine. In January 2020, Moderna announced development of an RNA vaccine named mRNA-1273 to induce immunity to SARS-CoV-2. Along the way, they partnered up with NIAID in March 2020, BARDA and Lonza Group in April 2020, and Catalent in June 2020. In March 2020, phase 1 human trials began. Preliminary results of the phase 1 trial was published on July 14, 2020. The vaccine was deemed safe in low doses. In 25th of May 2020, phase 2A clinical trial began. Phase 3 clinical trial began on 27th July 2020. In October 2020, the target volunteer number of 30,000 was reached. On December 30th, 2020, results from the phase 3 clinical trials were published. It indicated a 94% efficacy in preventing COVID-19 infections. Emergency Use Authorization, or EUA, was provided for the vaccine on 18th of December 2020 in the USA, 23rd December 2020 in Canada, January 5th, 2021 in Israel, and February 3rd, 2021 in Singapore. Conditional marketing authorization was provided on January 6, 2020 in New York, and temporary authorization was provided in January 12, 2020 in Switzerland. Dosage of the vaccine consists of two doses, each of 0.5 ml volume, 4 weeks apart by intramuscular injection. Common side effects of the vaccine include fever, fatigue, headache, muscle ache, pain at the injection site, flu-like symptoms. I will now hand over the presentation to Thomas.
Hello, I'm Tanzila. Now let's move on clinical trials. If we compare the randomized double-blind clinical trial methods of these two successful mRNA vaccines, we can see that there are around 30K or 40K number of participants. So these are phase 3 trials and at minimum age limit. For Pfizer, it is greater than 16 years old, and for Moderna's, it is 18. By intramuscular injection, two doses have to be taken. For Pfizer, the second dose has to be on day 21, and for Moderna, it is on day 28. The amounts per dose are similarly 30 and 100 micrograms, and the observation time is in span of 2 months after second dose. From the randomized double-blinded clinical trial results of Pfizer vaccine, if we observe the cumulative incidence, the red curve, we can see that the slope becomes almost flattened from the day 28, almost 1 week after the second doses, and the efficacy became around 95%. This is the chart of percentage efficacy of different subpopulations and with different coexisting conditions for Pfizer vaccine. The overall efficacy is 95%. Our data of interest here is older population greater than 75 years. For their case, the efficacy rate is very high. This is one of the reasons we have seen that Pfizer is recommended for older population in United Kingdom. Now, let's look at the Moderna practice vaccine clinical trial results. If we observe the cumulative incidence, the orange curve, you can see that the slope becomes almost flattened from the day 14 after the first doses. This is the chart of percentage efficacy of different subpopulations and with different coexisting conditions for Moderna vaccine. The overall efficacy is 94.1%. Our data of interest here is older population greater than 65 years. For their case, the efficacy rate is not very high, only 86%. So this vaccine is not well performed on an older age group compared to Pfizer vaccine. The side effect profiles of these two mRNA vaccines are satisfactory. Common side effects are injection site pain, fever, fatigue, muscle pain. These symptoms are very typical, normal, and expected after any vaccination. Very rare, severe side effect cases are anaphylaxis, a life-threatening allergic reaction, and it only happens to those who have a history of previous severe allergies. The most recent data from the CDC suggests that life-threatening allergies occurs about 2.5 cases per million for Moderna vaccine and about 4.7 cases per million for Pfizer. The excluded groups for taking these mRNA vaccines are people having severe allergies and bleeding disorders, pregnant and breastfeeding women, children under 16 or 18 years, immunodeficient states, cancer and HIV, and recent blood recipient donor. Some groups are very risky for vaccination, and some groups are not yet clinically tested, like children and pregnant women. Now, Shopin will continue the rest.
Hello, I am Shopin. Now let's talk about the advantages and disadvantages of the mRNA vaccine. Before commencing, we should know the COVID-19 vaccines designed by Moderna and Pfizer are the first two mRNA vaccines ever approved by FDA. So, while this new technology of vaccines holds immense potential, we are still not entirely sure about the long-term negative sides of giving mRNA vaccines to all humans. Now, let's talk about the good sides. mRNA vaccines can be designed faster than any other contemporary vaccines. It can also be produced faster because it is a genetic product. Strands of mRNA can be produced quickly once vaccine sequence is known. Secondly, in the case of COVID-19, both approved vaccines are showing very high efficacy of about 90%. Now, the negative side. The major problem from the conventional vaccines is the instability of the mRNA vaccine. To stabilize the mRNA, lipid nanoparticles are used. However, very low temperatures are still needed for long-term preservation of Pfizer vaccine. Hopefully, Pfizer have been working on the temperature problem, and according to recent data, the vaccines can be stored at normal freezing temperature for 2 weeks. There have also been some solutions, including dry ice for long-term preservation in remote areas. Now, let's see some major differences between the two approved mRNA vaccines from Moderna and Pfizer. Here we have annotated comparison about doses, eligible population, efficacy, and storage requirement. One thing to note here, while efficacy is almost same, amount of virus materials is almost 3 times in Moderna vaccine. Now let's talk about Bangladesh perspective. So time for some background. COVID-19 have been first detected on March 8, 2020. First death was reported 10 days later. The infection rate has been coming down from the end of the last year, and vaccination against the virus has begun from January 8, 2021. Let's have a detailed look at vaccination situation in Bangladesh. As of today, Bangladesh have vaccinated 3.11 million people, roughly 1.91% of the total population. The vaccine used in this program is Oxford AstraZeneca vaccine provided by Serum Institute of India. The vaccine provided is a non-replicating viral vector. It is also noteworthy that Ban-COVID, mRNA-based COVID-19 vaccines made by a local company, Globe BioNTech, has generated some hype, although the vaccine is still in animal trial phase. Now, what are the challenges faced to implement the FDA-approved mRNA-based vaccines in Bangladesh? Firstly, the storage issues, as it was initially touted that a very low temperature is needed for Moderna's vaccine, although that problem is now mostly solved by innovation and vaccine improvement. Second, the most major problem is production limitation, as Bangladesh has no production facility. To solve this, Bangladesh needs biotechnology industries. Finally, what is the need to do all this? Firstly, high efficacy. The amazing efficacy means lesser mRNA vaccines will be enough to enable herd immunity. And secondly, in the presence of emerging dangerous variants, the variation in vaccines give the population and country options to counter and reduce risk. Thanks for listening.